Hello, thank you for coming to my talk, and thanks to the organisers for inviting me to present. I'm going to talk to you today about where we stand in terms of cures for people living with HIV. I have no conflicts of interest. Now, there are rare examples of HIV cure, what we might term eradication or remission. For example, in 2009, Timothy Brown received a hematopoietic stem cell transplantation from a Delta CCR5 donor and remained virus-free until he sadly passed away very recently. 10 years later, in 2019, Adam Castillejo underwent the same procedure. And there are examples of post-treatment control and profound elite control which might be thought of as remission. Now, even though these are successes, they will not likely translate into scalable approaches for most people. However, they are very important proofs of concept that an HIV cure is feasible. Let's begin by asking a few questions about the reservoir. But most importantly, which is the HIV reservoir that matters, that will cause damage when antiretroviral therapy stops? The question, where is the reservoir, can be answered relatively straightforwardly. You can see, for example, this beautiful paper from Jake Estes and Tim Shacker shows that 99% of infected cells in someone on ART are, as you would expect, in the lymphatic tissues, most notably the gut, and these cells are the source of recrudescent virus on therapy interruption. What is the HIV reservoir? The majority of integrated proviruses are defective and therefore not replication competent. Shu Yu has recently shown that some competent viruses are integrated into gene deserts, areas of chromatin that are completely shut down. Other replication competent viruses may be found in CD4 T cells that are in such a state of deep quiescence that they are unrousable. So these three likely do not contribute to the reservoir that matters. What does matter is this replication competent virus that is found in a transcriptionally permissive site in a quiescent CD4 T cell that may become activated. Many investigators have asked, is there a phenotypic or transcriptomic profile of a quiescent cell containing replication competent HIV that we can identify and target? And indeed, CD4 T cells that are enriched in HIV may be found among proliferating and activated cells, and T follicular helper cells, cells expressing exhaustion markers. Less mature cells are enriched for HIV, but also expanded HIV clones are found in more mature cells. But I think it's unlikely that we will find a marker or even a combination of markers of a quiescent infected cell other than the proviral genome itself. And it's often been said that looking for that cell is like looking for a needle in a haystack. But I think it's more difficult than that, because it's like looking for a needle in a haystack that is made of needles. And how big is this reservoir? We understand that fewer than 1% of published virus sequences are classified as intact using various assays such as near full-length PCR. They should have no deletions, no frame shifts, inversions, or hypermutations. The problem is that a virus may seem perfect according to the assay you choose to interrogate it. It may make transcripts that you can detect by RT-PCR. It may make proteins that you can detect by ELISA. It may even be induced to make virions that are infectious in cell culture. But if that virus contains an easily missed functional error in LTR or VPR, for example, then the virus is not replication competent in vivo and does not contribute to the reservoir that matters. So all that I feel comfortable in saying is that only a tiny and difficult to quantify fraction of HIV genomes are able to produce replication competent virions in people on ART. But I'm not sure how important it is to know how big the reservoir is. Here is an example from Tim Henrich describing an individual who began ART on the day of exposure, who had no detectable reservoir that was thought to be less than 100 replication competent virions. Yet 8 months after therapy interruption, a single virus emerged. So what is important is not so much the size of the reservoir, but knowing how to remove it and stop it from growing. And as no biomarker accurately predicts post-treatment control or time to rebound, treatment interruption should be the clinical endpoint of cure approaches. Which brings me to clinical strategies for an HIV cure. Now, we can target the infected cell, which contains the virus itself, or we could enhance immunity to kill that infected cell. A single approach may affect multiple of these pathways, and I think it's important to stress at this point that combination therapy is almost certainly going to be necessary. So let's first consider very early ART. It's clearly not curative. I just showed you this example from Tim Henrich, and here is another study of more individuals. We're clearly up against a wall here. If we were to consider ART any earlier, we would be in the realm of pre-exposure prophylaxis. I'd like to state that very early ART can be curative in non-human primates, but as I've just said, not in people. And so from now on, I'm going to predominantly focus on studies done in humans. Latency reversal, or shock and kill, is an approach whereby HIV transcription is reactivated with a latency reversing agent, such that virus is produced as a result, and then the immune system, or the virus itself, will kill the cell, and it's done under the cover of ART to stop new infections. Multiple LRAs have been tested in vitro, and in vivo they show increased HIV transcription, however no decline in HIV reservoir size. Activation appears to be inefficient, tissue-specific, cell-type specific, and specific to the transcriptional state of the virus. More potent, less toxic LRAs such as SMAC mimetics show no change in reservoir size or delay in rebound. It seems that there is no kill of the infected cells, which are relatively resistant to death, and the virus, of course, has escaped the immune system. So the question is this a problem with these LRAs, or is it a problem with the approach in general? Have we reached a biological limit with non-specific LRAs? And perhaps we can add a degree of specificity to this approach. Which brings me to immune checkpoint blockade. Immune checkpoint markers are a variety of receptors on many different immune cells that act to dampen the immune response. Immune checkpoint blockade non-specifically enhances immunity, but within that immunity there is of course HIV specificity encoded in HIV-specific T cells and B cells. PD-1 and CTLA-4 are expressed on exhausted T cells in HIV infection, and they have been shown to suppress this immune response. Immune checkpoint blockade in vitro and in vivo also reverses HIV latency. And HIV is enriched in PD-1 positive cells in blood and lymph nodes from people on ART. So immune checkpoint blockade acts both to reverse latency and enhance immunity. However, results from clinical studies to date are variable and generally not encouraging, although I would say it is still quite early days. Here's an example of a phase 1 clinical trial of anti-PD-1 alone or with anti-CTLA-4 in people living with HIV on ART who had cancer. Now, HIV transcription increased in the recipients of anti-PD-1 and CTLA-4, and what you can see here is that there was a significant decrease in virus outgrowth in 2 people. So it's not as encouraging as we would have hoped, but remember, it's a start. We can try to enhance immunity specifically using vaccines, and in fact many therapeutic vaccine trials have been completed over the past 20 years using many different vaccine modalities. They have been safe, generally immunogenic, but of no benefit in most studies and only modest benefit in a few studies. We understand some of the reasons why therapeutic vaccines may not work. There are pre-existing immunodominant responses. Virus epitope escape has occurred. There is lack of potency of the vaccines, and this is in the setting of overall inflammation and immunosuppression, as well as there being immune-privileged tissue sanctuaries. So there are many barriers to therapeutic vaccine efficacy that we understand, and indeed some of these may be overcome, for example, using immune checkpoint blockade. I'm showing you here the result of a trial that was presented at recent CROI, in which 45 individuals who began ART in acute infection were recruited to receive a vaccine consisting of DNA and MVA or placebo, were then rolled over to receive chimp adenovirus MVA vaccine or placebo, which was followed by a therapy interruption, and then ART was restarted. Now you can see in these results here that in the individuals who did not have protective MHCs, that there was a modest effect in that therapeutic vaccination induced a slightly lower virus set point. Which brings me to broadly neutralizing antibodies specific for HIV, many of which have been discovered over the past few years. I'm showing you at the bottom the names of these antibodies, and at the top I'm showing you the breadth of envelopes that are neutralized, a panel of 208 HIV-ENV pseudoviruses. The y-axis shows you the potency, and as you go down, the potency increases. So CD4 binding site antibodies are broad and potent. Those specific for glycans are less broad, but can be much more potent. The EMPA targeting antibody 10E8, very broad, not so potent, it met with some manufacturing difficulties. And when we combine specificities with combinations of antibodies or trispecific antibodies, they can be very broad and very potent. And this study in which 2 potent bNAbs were administered to chronically suppressed HIV-infected people showed that those with bNAb-sensitive virus remained suppressed for 15 to 30 weeks during a therapy interruption. If we consider the breadth of coverage of combinations of antibodies or trispecific antibodies, and we put it in the perspective of the recent antibody-mediated protection or AMP trial, where at an IC80 of 1 microgram per mil, VRC01 only gave 29% coverage. If we look at the table on the right, then the percent viruses neutralized for any of these antibodies on their own is 49%, 63%, and 83%. The theoretical combination where only one antibody needs to be active against an envelope is 94%, if 2 active, 74%, and if 3 need to be active, 27%. The coverage for the trispecific antibody is 81%, considerably greater than that for VRC01. And clinical trials of these combinations are currently being performed by Boris Yulg. One study has been completed, and another study consisting of 6 monthly injections in ART-suppressed people with a therapy interruption after the first dose of antibody is enrolled, and I'm really looking forward to seeing the results of that study. Now, an exciting way to deliver these bNAbs is to use the genes that encode them. One could perform ex vivo modification of B cells, or more simply, one could deliver the genes using viral vectors such as adeno-associated virus AAV. The advantage is that transduction occurs in vivo after IM administration. The transgene persists in the nucleus and is stable in post-mitotic cells. And this experiment using AAV to deliver monoclonals performed in non-human primates showed sustained suppression of viremia after AAV delivery of bNAbs. Here are the results of a study presented by Joe Casazza at the recent CROI, where AAV vectors were used to deliver VRC07 to 8 HIV-infected people on ART. You can see the results in the graphs on the right. 5 of these 8 volunteers produced functional VRC07, which started 4 to 6 weeks after AAV administration. 4 had VRC07 concentrations at or near maximal for 1 to 1.5 years. And this is remarkable. This is the first trial to show the induction of a difficult to elicit broadly neutralizing antibody in humans by a therapeutic intervention. I mentioned at the beginning that I thought combination approaches are very much likely to be in the future of HIV cures, and there are a number of approaches combining LRAs with various immune interventions. And I'd like to draw your attention to this one from UCSF, sponsored by AMFAR, where the LRA is a TLR9 agonist, which also acts as an adjuvant for a vaccine, and that's given in combination with 2 bNAbs. That study is enrolled, and at the moment it's in the therapy interruption phase. Many other trials combining bNAbs with immunomodulatory agents are underway. However, significant effects on the HIV reservoir remain to be shown. Now, I said at the beginning of my talk, and many people say the same, that the examples of Timothy Brown and Adam Castillejo are proof of concept of an HIV cure. However, that's not quite right, because more specifically, they are proof of concept of an HIV cure by gene therapy. That is, the targeting of the host genome to disable expression of particular genes. One could disrupt the HIV proviral genome itself, or one could remove something that HIV needs, such as CCR5. And the tools available to do this include CRISPR-Cas, nucleases, shRNAs, and so on, and one can manipulate the cells ex vivo or deliver the agent in vivo. And I'd like to show you some data from a remarkable study recently posted to bioRxives by Rafik Sekali and his colleagues, in which 15 people with HIV, fully suppressed on ART, underwent leukapheresis, from which CD4 T cells were enriched. Some of them obviously express CCR5. These cells were then treated ex vivo with a zinc finger nuclease that physically cuts the CCR5 gene. And what you get is a product consisting of CD4 T cells, some of which cannot express CCR5. These cells were then reinfused, and a therapy interruption was performed 6 weeks later. Importantly, this immunotherapy is minimally invasive with no severe adverse events. It's much more accessible than hematopoietic stem cell transplantation, and there is no need for compatible donors and no risk of GVHD. And the results show that 5 individuals partially controlled virus after treatment interruption, so we may call them post-treatment controllers. They had decreased virus reservoir size in terms of integrated total and intact provirus, and they showed persistence of CCR5 edited CD4 T cells. Interestingly, one individual controlled virus load for 5 years after infusion, and it's thought this may be due to a small replication competent reservoir, persistence of CCR5 modified cells, and increased HIV specific T cell immunity. I find these results very encouraging. So where do we stand, and where are we going in terms of an HIV cure? I think I've made it clear that it's still very early days. We have to understand there is a whole spectrum of how people living with HIV fare on ART. Some may be well with no problems of adherence or access. Others may not be doing so well for many various reasons. And within that spectrum, we must be able to offer a range of scalable and safe options. From a true cure with no virus, to various remission strategies, to the understanding that some individuals want to be left alone and are perfectly fine and would like to remain on their current regimen. Understanding the biology of persistence of the quiescent HIV reservoir is critical to discover targets. We have to test multiple approaches in non-human primates and humans, and by multiple I mean combinations and lots of them. We must also not be afraid to call it quits for an approach which is going nowhere. This will likely be a long, bumpy, and iterative process. The first cure will not be able to compete with ART for most people. But I have no doubt that we will get there, because that's what we do as scientists, as doctors, as everyone associated with this effort. We make progress. It's our job. I'd like to thank you all for listening. I'd like to thank everyone in my lab, my collaborators, and especially Sharon, Steve, Rafik, and Mirka for help in putting this presentation together. Thank you.